and start with whatever doubts you have. I will pick it up from there as we wait for others to join. Can anyone hear me?
Yes, sir.
Good. All right. So I have a direct question here. So please, could you explain active transport? Okay, good. So yes. So to talk about active transport simply is one of the means by which substances are transported in plants. Okay. Now, to break it down, it's one of the ways by which substances enter the cells of living organisms. Okay. So let's say you have glucose outside the cell, but you need the glucose inside the cell. How do you get the glucose outside the cell to enter the cell? All right, good. Another example is you have water outside the cell. You want water inside the cell. How do you get the water into the cell? Okay. You also have another example. You have oxygen outside the cell. But you all know that we need oxygen inside the cell so that the oxygen can be used for a metabolic reaction, respiration. You all know that. So how does the oxygen enter the cell? So we understood from chapter 3, I guess, movement in and out of a cell, that there are three ways by which substances move in and out of a cell. The first one we talked about is diffusion. That mainly has to do with gases. The second one has to do with osmosis. That has to do with water movement, water molecules entering the cell. And the third one has to do with what we call active transport. Okay. So now coming back to your question, what then is active transport? We've laid down a basic understanding that active transport is one of the ways that enter the cell. So in active transport, what we are looking at here is that how do substances that are within an area of low concentration get to an area of high concentration? How is that done? So we understood from the diffusion point of view that for a substance to move, it needs to move from a higher concentration to a lower concentration. That is diffusion. Okay. But in active transport, we need the substances that are at the lower concentrated region to move towards where there is a higher concentration of that particular substance. For example, we have minerals in the soil. So if you have minerals in the soil, what can we say? Minerals in the soil, in this case, is needed in the plant cell. But how did this mineral get into the plant cell? This mineral gets into the plant root hair cell by what we call active transport. One, they dissolve in the water, they are picked up. But then they also have a way of entering into the cells when they are in the plant. And so how do they move? Inside the plant, there is high concentration of minerals, but we still need the little that is to enter the plant. So in active transport, we say the minerals need to enter the cell by the process we call active transport. Why do we say by active transport and not by diffusion? One, the first reason is that by active transport, because the substance, which is the mineral, is moving from a lower concentrated region to a higher concentrated region, which is inside the cell. Two, now because it's moving from a lower region to a higher region, it means that it will need energy. Just like if you have a giant and you are small and you want to maybe beat the giant or push the giant or whatsoever, you need an extra energy to cause you to push it. Let's say the thing you have a slope, you need to push it upwards. All right. In that case, you need to exert more energy to push the thing upwards. But if the slope goes down, you need energy. No. Once you leave the thing will slide down. Uh, so this is what we are talking about. This, this is what we are talking about. Please just a minute. All right, I'll continue with you guys.
No.
All right, very sorry for that. So let's continue. So um, active transport, as I was saying, one, the substance is moving from a low concentrated region to a higher concentration. Two, it needed to use energy because it's moving from low to high. So there is a need for energy to push the substance through the cell membrane to enter into the cell. And now, how would the energy be used to cause the substance to move into the cell? Now, we said within the cell or around the cell, we have what we call cell membrane. This cell membrane allows substances to control what enters and leaves the cell. Within this cell membrane, we have what we call carrier proteins. So these carrier proteins are more or less like the gate to allow the substances to enter. So these carrier proteins, they need to adjust, open up to the outside of the cell so that the mineral can enter and so that it will reshape itself, manipulate it for the mineral to enter the cell. So the reshaping of the carrier protein, that kind of reshaping, that kind of movement is what causes the substance to enter the cell. So if you should ask about what active transport is, that is, I'm not using diagrams because you already have all the diagrams in your book. So just making reference to what. And so if you should ask what are some of the examples of active transport in living organisms, in humans, you need glucose or you need substances, nutrients to enter into the cell and test back the transport. You can also talk about implants to minerals to enter the cell. There's a cell by active transport. If I haven't answered you, or if you still have a doubt regarding active transport, you can just drop the chat. Thank you for that question. Any more question? Anyone have a question? A good one is, could you please go to part A and part B of question on page 145? Are you referring to the course book? Please come again. If you are referring to the course book, let me know. Okay, so coming back to that, I think there is also another question. After you explain this, I don't understand. All right, so so let me just share the screen of what the person shared. Let me see. All right. So, I think your question has to do with the apparatus. That's question 7 of the MCQs. The apparatus shown is used to demonstrate osmosis. All right. Dialysis tubing acting as a partially permeable membrane. Okay. So the mass of the dialysis tubing and content was 11.2 gram at the start of the demonstration and 9.4 gram at the end. Which solution would cause this change? All right. So it said the mass of the dialysis tubing and content was 11.2 at the start. So if at the start the content was 11.2 and they are saying and 9.4 gram at the end. So what has happened? Has it lost mass or it has gained? So you're looking at the values, you can see that 11.2, 9.4, there is a decrease. All right. So if there is a decrease, then it means that substances moved out of the dialysis tubing. All right. And you know that it can only be water molecule. So it's by osmosis. So substance moves from K to P. All right. So the question says, which solution would cause this to impact? So we said K, solution P. Yeah. So we have solution P and solution Q. So the basic understanding is that solution Q is going to have low water potential. Solution P. That is what will cause water to move out of the water Q to P. And so with that understanding, you can now get the question answered. So Emma, come back with the answer from this little explanation. Come back with the answer. Let me just check whether you understand what I just said. Okay. Yes. Okay. So back to your question from the course book. Now, notwithstanding, it's not only just about chat. If you feel like you want to ask a question, fine, feel free. Just unmute yourself and go ahead with your question. It's not only about chat. But if you feel like leaving the chat, all right. All right. So, Johnny, let me get my course book and I'll get back to your question. Okay. I needed to see what you're actually talking about. All right. Good morning, sir. So, please, could you explain the terms independent and dependent variables and how do we use it in an experiment? If you have a question on it. All right. Good. So, just picking it up from a simple point of view, you have a mom, you have a dad. Okay. So in this case, you are depending on your mom and your dad for everything. So your outcome is dependent on your mom and your dad. Okay, your outcome is dependent on your mom and your dad. So in this case, you cannot tell your dad or your mom to sit at home and not go to work. But your dad and your mom can tell you, sit at home, don't go to school. And you have to sit at home, not to go to school. So what are we saying here? In this case, you are on your mom, and so you become the dependent variable. Okay? But then your mom and your dad, they don't depend on you. So they are independent. They don't depend on anyone. So they are independent. Okay. So in this example, coming back to the classroom or the lab, what we're trying to say is that in the lab we have variables, okay, factors. Now, some of the factors depend on others. So the outcome of a factor depends on others. All right. So in this case, let's look at something. Let's see a typical example. So we have what we call, you are recording the time taking for water to move into the cell to a certain mass. All right. So what is your interest? What are you going to record at the end of the day? You are going to record the time taking. All right. So the time should be varying per your experiment. So that is your result. So result is always dependent on certain factors. All right. So if the factors, let's say the content, or let's say the substances, if it's potato, pumpkin, uh, cassava and co. Those are the, let's say, content or the substances. The time it takes, the time is what is varying. That is of what is of interest to you. That is what you are measuring. Okay. So what you are measuring, your results, your outcome becomes the dependent. What is it depending on? It's depending on those factors that are giving you those results. So if you talk about independent dependent variables, just look at the variables and ask yourself which ones are the results. Which ones am I getting at the end of the day? The ones, which ones am I measuring? All right. So the ones you are measuring, the ones that keeps changing, they are dependent on the ones that are fixed. Okay, that did not change. So get it from this understanding point of view. And the dependent variable is always the one you are measuring. It's always the result. Okay. So that is basically on that. If you still don't get it, let me know. Okay. So put a
Thank you, sir.
typical question to work and try. If you don't
Sir,
still
I didn't
get it,
get it.
just no attention. You don't get it.
No.
Okay. So going back again to what is dependent and independent variable. So the one who said he didn't get it, you do you know how to cook? Unmute and talk. Let's have a discussion. Do you know how to cook? I want you to get it. So let's have a discussion. The one who said he didn't get it, do you know how to cook? The one who asked the question, you understand? Jairus, you get it. Hello, can anyone hear me? Can anyone hear me?
Yes, sir.
All right.
Sir, I can hear you, but your voice is like breaking.
Oh, sorry. Okay, so what I'm asking is that, do you understand the dependent and independent variable?
Yes, sir.
All right, good. That's okay. I think there was a question on would the structure of the tooth come in the exams. Well, it will come in the IG, so you just have to learn. I mean the IG, yeah, 10 IG. And you know whatever we are doing now is part of the 10. So if you don't see it in this current exam, just learn for it. Learn and make sure you keep it in mind for subsequent exams. All right. So please, the person who has the question, I can't tell whether it's coming or not. All right. But the fact that I've taught you in class should give you an indication that you will access on it. So please learn, and it's not a big thing. Learn the structure, learn the various parts of the tooth, and get to know their various functions and how they look like. Okay, just that. Just keep those info in your mind. So if you don't even get it in this exam, subsequent exams, you see that you'll be able to answer them. All right, thank you. More questions. Translocation. Okay. So if you talk about translocation, simply the movement of sucrose and amino acids. All right, in plants. So the transport of sucrose and amino acids in plants. We know transpiration is the loss of water. So it has to do with what? Water, the loss of water. But translocation has to do with the movement of nutrients. All right, that is sucrose and amino acids all around the plant. So we understood that it moves in two different directions. And the vessel it moves is what? Is a phloem vessel. So if glucose is produced in the leaf, we know glucose cannot be transported as glucose, but it's changed to what? Sucrose, and it's transported as sucrose. So if glucose produced in the leaf is needed in the roots to undertake respiration, what will happen is that that which is produced in the leaf needs to be what? To be moved from the leaf down to the root. That mechanism of movement is what we term as translocation. And translocation, we look at source and a sink, where it is produced or where it is released. We call it the source, where it is deposited or where it is used or where it's stored. We term that as the sink. So that's about translocation, and it happens only in plants. Okay. Any more questions? Any more questions? I'm trying to locate your question. I'll be glad if you can get a picture of what you're actually talking about. Because all right. Any question?
No.
Okay. Question 9. Exam style question. Granny, can you leave behind the topic? The topic. Okay, I see chapter 7. Okay, thank you. I'm getting to the question. Okay. So I see your question. Question 9. The photograph shows a session through two villi. from the human digestive system. Yeah, and that is the villi. So we have the smaller one. We have one at the left, one at the right. So what is the question saying? The question is saying, could you please go through part A and part B of question? Okay, so part A says, name the part of the human digestive system in which villi are found. Okay, so the parts where villi is found, you already know that's one question. All right, that's more interesting. Name the part of the villi labeled A and describe its function. Okay, and what's the question again? Okay. So we looked at A. When you looked at A should be the epithelial cells. Okay, A should be the epithelial cells. We know the epithelial cells are around the villi. So A should be the epithelial cells. And what is the function of the epithelial cells? The epithelial cells, we understood that it secretes motility. Okay, and motility digest the maltose that has been digested by the amylase in the mouth. Okay, so epithelial cells secrete maltase, and the maltase will digest what? The maltose to glucose so that it can be absorbed. All right, so that it can be absorbed into the blood. This is the cell B secretes mucus. Suggests the function of mucus is part of the digestive system. All right, so you already know mucus. Mucus is basically for, in this case, mucus is for lubrication. Mucus is for lubrication. What is lubrication? Reduction of friction as the food is moving down through the alimentary canal. So, yeah, that again is the part, the cell level B. So they said the cell level B secretes mucus. So you already know what B is. B should be the goblet cells. All right. The part labeled C is the blood capillary. So they've already told you what C is. So A is the epithelial cell, B is the goblet cells, and C is the blood capillary. Then name two nutrients that are absorbed into the blood capillaries in the villi: glucose, amino acids, the rest. Some nutrients are not absorbed into blood capillaries. Name the structure into which these nutrients are absorbed, and that is the fatty acid and cholesterol. We know there is a presence of lactose, all right? Lactose, and lactose helps the absorption of fatty acids and cholesterol. Okay, good. Thank you. All right, you're welcome. You're welcome. All right, so now let me take you through the entire exam structure, what you should expect in general. What you should expect in general. As I told you earlier, you have 40 MCQ questions. We've done a lot of MCQs. MCQs play a major role in your exam. And I kept telling you, you shouldn't play with the MCQ because it forms 40% of your marks. 40. So if you manage to get 38 over 40, then for sure you are already getting close to your A star or your A. or even B, then the little effort you put in your section B, which is the theory and the practical, you are good to be in the A corner. All right. And all the MCQ questions that you are going to see in your exam, we've already done them in class. If after exam and you realize there is an MCQ question that we've never talked about in class, you can draw my attention to it, and I apologize for that. But all I want to tell you is that everything on that exam paper has been done in class. Okay, so please go over the revision worksheet for the MCQs that I've already given you, the ones we've done so far. That is about the MCQ. Good. We move to the paper 2, which is the theory. Paper 2, the theory. Now, you need to give precise answers. When I say precise answers, I mean you must be specific to what you are talking about. Don't beat around the bush, use biological language, to answer questions. Okay, don't go be writing planes. I've taught you how to address questions with command words. So if they ask you to explain, you have to give the reason why a kind of phenomenon is happening. Okay, you have to give a reason to why something is happening. If you are asked to describe, what it means is that just talk about what you see. What are you seeing? So if you're asked to describe, let's say, a graph, you have to talk about what you see. So talking about a trend, let's say, is it increasing or decreasing? So that is the description you need to give. So, okay, so and so decreases while so and so increases. Okay, oh, okay. An increase from 0. So and so to so and so. You get it? So add values to buttress the kind of description you are giving. All right, take note of that. Also, I also want to tell you with where you have to match. Okay, you have some questions you need to match. If you are not too sure, don't start.
Yeah.
Can anyone hear me?
Yes.
Okay, so a quick one. Let's go on. The rest to join. So still talking about the exam structure. So as I said, if there is any mark you need to do, make sure you are sure before you use a pencil and a ruler to match neatly. Okay, don't match using a pen. Then later you cancel the match you've done, then rematch to a different one again. When it happens like that, it means you put me in a tight corner to decide which one I should mark correct or wrong. And with that, you will mark wrong. So make sure you are sure of your answers before you do it. You use your mark, do the matching. And if you also have a box, if you also have a box and you need to write a letter inside, let's say P or Q or R, make sure you know what exactly you are writing in that box. Don't write two or more letters in a particular box that requires for only one letter. Take note of that. So you can first use a pencil. If you're sure, you change it to pen. All right. So you have simple questions on the human nutrition. When a person eats, how food runs through the alimentary canal. Okay, the process it moves through the alimentary canal. Okay, it starts from here and what it moves through this side and it ends here. You should be able to go through that. And we talked about the substances that are released, hydrochloric acid and its importance, the enzymes and their importance. We talked about all those things. Okay. And we can also talk about enzymes. So enzymes, we did enzymes previously, the previous uh, semester or previous term. So make sure you go over the enzyme, know what enzyme is, and you'll be able to identify the enzyme, its active sites, substrate, product, enzyme substrate complex. All these things we looked at them. All right, good. And why you think a particular thing is an enzyme. Also, we looked at the level of organization of an organism. Start from the cell, built up to the tissue, built up to form the organs, form the organ system before the organism. You should be able to. And if you take the leaf as an organ, you should be able to tell the structure of the leaf, the tissues. It has what we call the waxy cuticle and the rest. All right. So take note of that. All right. Can you guys hear me?
Yes, sir, we can hear you.
All right, good. Nasara, please, can you mute yourself? Thank you. All right, then we can move on to transport implants. All right, transport implants, we looked at what we call transpiration. We looked at how substances are absorbed into the plant, how they move up. We looked at the vascular band, the xylem phloem, and we look at their various functions, what they carry, and their petty descriptions. All right, so we looked at that too. And also, we also looked at the physical digestion, where we talked about the teeth, and we also talked about how food moves down by peristalsis, all those things. So the tooth, we looked at the various parts of the tooth, and to be able to identify a particular tooth and what it does. All right, so take note of that. Once again, I talked about enzymes, take note of that. I stressed more on the types of digestion, chemical and physical, and of what importance physical digestion is, chemical, all those things. I talked about that in class and with this series of exercises, assignments. So make sure you go through those also. If you are given the structure of a human digestive system, you should be able to know, okay, the various parts, their positions, where they are, and their functions. This is very important. You should know that the stomach is for so and so. You should know that the mouth is for so and so. You should know that the anus is for that, is for this. You should be able to know that. Also, we also looked at movement in and out of the cell. We looked at osmosis. We looked at diffusion and active transport. I think the last time we did one practical in the lab where we went to look at substances entering water moving into the potato cell, right? So we looked at that kind of experiment. Yes, the results we got didn't really make much sense, but just revise around what osmosis is and get to know what will happen to a plant cell when it's left in what a concentrated solution. Look around those areas. Talk about the rigidity of a plant, what it means. Good. Now you have biological drawing. You should make sure you pay attention to all the rules that govern biological drawing. All right. You have a nice picture there that you have to draw. Just make sure you read the question. Let me stress on this. A lot of you don't read the question, especially that comes with the biological drawing. As soon as you see the drawing, you just start drawing. Make sure you read the question. The reason for this particular question is that if you skip the question and just start drawing, you mess up with what you draw and you lose marks. So make sure you read the question. Get to know what exactly amidst the entire drawing, what specifically are they asking you to draw, and follow the biological drawing rules, and you should be able to do the basic calculation. If you're asked to find magnification, you know how to do it. If you're asked to find actual size, you know how to do it. And yes. And so once again, I'm stressing it. Make sure you read instruction with regards to the biology practicals. Make sure you read the instruction. If you don't read the instruction, you will find yourself wanting. Follow the instruction and you get everything right on point. Okay. And there is one question that has to do with your analytical thinking ability, how to think critically. Okay. They are still the questions are also still within your zone. I mean within what you have been taught in class. So an experiment is carried out. You need to reason around the question and give an answer. All right. So that one I didn't talk much about it because it is to test how you think critically. And once again, it's not far fetched from what we have. Okay, we talked about how to find average of experiments. If you run experiments, we understood that we repeat experiments so that you can make the results reliable. So after you've repeated the experiment, you needed to find the average. And how do you find the average? You add all the values you got, then you divide by the number of times you repeated the experiment. That gives you the average. All right. If you are leaving your average in one decimal place, all the values should be in one decimal place. If you are leaving it in two decimal place, all of them should be in two decimal place. So take notes of that too. All right. Now we also looked at optimal temperature, what it does to enzymes. All right. On that note, I will say we've come to a fruitful end of the doubt clearance session. I'll leave the room for anyone who have any question again. If there is no question, all right. So with biological drawing, we have only one biological drawing. Okay, only one biological drawing. Only one biological drawing. But what I mean is that you must read before you start. Make sure you read every question before you start. Good. Any more? All right. Any more? Okay. So as I told you earlier in class, that your worst grade should be an A in biology, especially regards to this paper. Okay? It should be an A. You have done so much from the very beginning of the term to this point in time. You guys have been so good. You have been so oh sorry. Check like I went mute. All right. So as I was saying, almost all of you have been so cooperative and so studious and throughout the term. I'm very grateful for that and I appreciate all that you guys have done. And I know it's gonna reflect in your exams. So let's go for an A star. If one or two mistakes come in, then you remain at A. Okay. I know you guys can do it. Those of you can get the A star. Aim for all the marks. Don't say because I'm getting 90, that's okay for me. If you have the ability to aim for all the marks, go get it. All right, it's possible in biology. Wish you the very best once again. Thank you. That's been a good and a fruitful academic year. I wish to be smiling when marking your paper. Thank you once again. If there is nothing to address, I request to end the Zoom meeting now. If there's anything to, you can just unmute.